The very nature of autoimmune diseases and processes is that your cells meant to protect you attack you instead. When we examine the inner workings of autoimmune processes, T cells and B cells are the driving forces behind what happens within our body. From type 1 diabetes to lupus erythematosus, all autoimmunity stem from an error in checkpoints for immune cells. For example, B cells that are tested in the bone marrow and the spleen for autoreactivity are usually eliminated if they exhibit a reaction to the self-antigen presented. If they aren't eliminated, they will continue to the next checkpoint and eventually be used in the lymphatic system, which is a huge danger warning. Because of these self-checkpoint errors, the autoreactive immune cells can be activated in a couple of ways. One way is molecular mimicry, basically molecular fraud. In molecular mimicry, viral proteins are too similar to self-proteins, or the receptors on your cells. B cells and T cells get confused because they look too similar and end up attacking both the viral proteins and your self-proteins. Molecular mimicry is made possible by the works of bystander activation in dendritic cells. Think of dendritic cells as the security cameras within a cell, with column B and T cells in case of a viral break-in. But if the surrounding tissues of a cell are damaged, dendritic cells mistake self-antigens for viral antigens, causing the molecular mimicry reaction. Epitope spreading is what exacerbates the autoimmune response. The immune response that resulted from initial cell damage and inflammation causes more self-damage, which creates more self-antigens and more autoreactivity from B and T cells. It's a never-ending cycle. So, how do we fix autoimmunity? The main treatment currently used for autoimmunity is immunosuppressant drugs. Small molecules inhibit the activation of adaptive immune cells and target them. However, adaptive immunity depletion needs repetitive infusions to maintain control, and it can lead to chronic immunodeficiency and risk of infections. Enter CAR T cells. CAR T cells are T cells with the CAR, or chimeric antigen receptors, attached to them. These cells recognize B cells that haven't quite been finalized in the checkpoint process and eliminates them. This gets rid of most of the possibility of B cells causing reactions with the body's self-antigens. To introduce the CAR T cells to the human body, white blood cells are separated out of the blood and brought to a lab. There, a virus is inserted into T cells and new genes are produced, causing T cells to make special receptors on the surface. Finally, the white blood cells are returned to the patient's bloodstream. Although CAR T cells are known for their cancer-fighting ability, they are showing significant promise in terms of blocking autoimmunity, and I can't wait to see where they go next.